Hi, my name is Amelia Grace, and then today we'll be discussing the determination of a gender of an embryo based off of the hormones and genes it contains. So, to start off, there are three different sexual organs that are very prominent in determining sex. One are the gonads, internal genitalia, and external genitalia. Now, the gonads are in males are the testes and they produce sperm, and in the females they are the ovaries and they produce ova, which is an egg. Now the egg and sperm are also known as gametes. So eggs and sperm cells are haploid, which means they're made up of 23 chromosomes. There's 22 pairs of them within the 23 chromosomes, and then there is one unmatched pair, not even a pair actually, it's just one sex chromosome. And now a zygote will receive the sex chromosome from the mom and one from the dad. So now if we're talking about autosomes, which are body cells that are not sex cells, there are 46 chromosomes. They're known as being diploid because 22 are matched, or they're known as also homologous pairs, and then there is one set of sex chromosomes. Now, the sex chromosomes, which is on the 23rd pair, are a determinant factor of whether the zygote will be a male or a female. So let's look at the first picture right here. You can see that there's chromosomes and there's pairs, and then on the 23rd pair, which are the sex chromosomes, there are two X's. The two X's indicate that the zygote will be a female. Here we have an X and a Y. Now with the Y present, the zygote will be determined to be a male. Now over here, we have an XX and a Y. However, the Y is still present and the individual will be a male. Now let's look if there is a missing chromosome. So here we have an X with no other pair, it's unpaired. There's an X missing or a Y missing. This individual will still be able to live, however, it will suffer from Turner syndrome. And over here, there's no karyotype if there's a Y chromosome present, but no X chromosome present, because the X chromosome contains essential genes needed for an individual to live. Also, the X chromosome is much larger than the Y chromosome. Okay, so in the early stages of embryonic development, it's very hard to determine whether the embryo will be a male or female until the 7th week, because all of the structures, male and female structures, have not yet developed. So the tissue is known in the embryo before the development is known to be bipotential. The sex cannot be determined yet. So the bipotential in the different sex organs, such as the bipotential internal genitalia, include Wolferian duct and the Mullerian duct. The bipotential external genitalia includes the genital tubercle, urethral floats, urethral groove, and labioscrotal swellings. Now, the bipotential gonads has an outer cortex and an inner medulla. Now, as sexual development occurs, the outer cortex will become the ovarian tissue if it was a female, and the inner medulla would become the testes if it was a male. So a very huge determining factor of whether the bipotential tissue will form into male organs is the SRY gene, which is also known as the sex determination region of the Y chromosome. So this SRY gene acts on the Y chromosome. If the SRY gene is present, the bipotential tissue will form into testes. And if it isn't present, then the bipotential tissue will turn into the female ovaries. So this slide shows an overview of how the S or Y gene acts on the Y chromosome. And this person is obviously going to be a male because the S or Y gene is present, and it develops the testes. After the testes develop, there's different cells that produce different hormones that are important in producing the male reproductive organs and structures. So once the testes are produced, the Sertoli cells release anti-Mullerian hormone, and that causes the Mullerian duct, which was going to become the female, which would turn into the female genitalia, to dissipate. Now, the Leydig cells they secrete androgens, and testosterone and DHT are dominant steroid hormones. Now, testosterone converts the Wolffian ducts to become the epidermis, vas deferens, and seminal vesicles, which are male structures. And then later on in development, the testes from the abdomen will grow into the scrotal sac. So DHT is very important because it also helps the external genitalia to develop, as well as the prostate gland. Now, if there is no SRY gene present, the gonads develop into ovarian tissue, and with no AMH present, the Mullerian ducts develop into the vagina, uterus, and fallopian tubes. Without testosterone, the Wolffian duct degenerates, and without DHT, the external genitalia takes on female characteristics. Okay, so this picture depicts the different genitalia developed at different stages of embryonic development. So on the left is a female, and on the right is a male. Now the genital tubercle, which was part of the bipotential external genital, turn into the clitoris if it's a female, and then if it's a male, it forms the glans penis. The urethral folds and grooves turn into the shaft of the penis if it's a male, and then it turns into the labia minora, the opening of the vagina, and the urethra if it's female. Now the labioscrotal swellings form the labia majora, and the labioscrotal swellings in a male would form the shaft of the penis and the scrotum.